Okay. I don't know if you can get the top of my head with that camera. So I want to thank the organizers very much for inviting me to such a diverse and interesting meeting. My topic for today is understanding and re-engineering nucleoprotein machines to cure human disease. It was carried out in the Nanomedicine Center for Nucleoprotein Machines, which is a consortium of universities in the US and Europe, funded by the NIH Roadmap for Biomedical Research. So, nanomachines, natural nanomachines, they fall into four categories: those that generate mechanical force, those that generate chemical gradients, those that carry out chemical transformations, and finally those that carry out information storage and transfer. And nucleoprotein machines are concerned with the last of these functions, fundamental functions: information storage and transfer. They are characterized by an ability to self-assemble within the cell nucleus. They carry out processes of DNA replication, copying of DNA into RNA, translation and processing of RNAs. The particular machine that we work with is the machine that's concerned with the repair of DNA double-strand breaks, and I'll talk about those more in a moment. But first, my theme for today is the development of simple nano devices. That is, devices with one or two components that interfere or give new functions to these complex nucleoprotein machines. So this concept's a little different than what Ralph Merkel, for example, talked about yesterday, which is this sort of bottom-up engineering of machines with new functions according to a complete design. And it's more in the nature of working with this evolutionary process of biology that we just heard about in Dr. Bloomberg's talk, the idea that cells really have a lot of parts lying around that can be adapted for new purposes. There are all kinds of examples of this in biology, and I'll give you an example at the end of my talk today. The particular machines that we work with are those that repair DNA double-strand breaks, and I'm going to give just three examples today of what we're doing with these. First, we're modifying the machines in a way that we can see them in the optical microscope by introducing bright photostable probes into components of the machines. Second, we've developed a way to introduce a modified single-chain antibody into cells to inhibit the function of nucleoprotein machines. And third, we're working on a way to introduce new functions for gene correction. So first, in brief, introduction to DNA double-strand breaks. How are they formed? Why are they important? Why did they inspire us to work on these machines? Well, ionizing radiation and recombination nucleases are the natural sources of DNA double-strand breaks. In ionizing radiation, a particle or a photon moves along a defined track, transfers energy to water and other molecules that it encounters, and introduces chemical damage on the nanoscale in a very structured way. So that a very small amount of energy can do a lot of biological damage. And here's an illustration of a track intersecting a molecule of DNA and introducing clusters of damage on the two strands of the DNA that will result in a physical interruption of this chromosome. So most kinds of DNA damage the body handles quite well. All of you at this moment, there's a lot of DNA damage and repair that's going on in your body, 100,000 damage events per day, lots of metabolites of DNA excreted in everyone's urine. Double-strand breaks are different because they affect not just one gene. After all, we have two copies of most genes. Most of the genome doesn't do anything anyway. But a double-strand break potentially affects thousands of genes, everything that's distal to that break on the chromosome. So cells really have an elaborate way of dealing with these breaks. They're gravely dangerous. A few dozen breaks are all it takes to kill a cell. This is a minute amount of energy being transferred, but it's being transferred in a structured way. So how do cells respond? Well, they have ways of tolerating this damage. breaks are mostly dangerous when the cell divides. So cells have an elaborate mechanism for sensing when these breaks occur and going into cell cycle arrest. Cells can die. Now, clinically, this is very important. That's how radiation therapy works, is kills cells by introducing DNA double-strand breaks. Or they can repair them. And there are two pathways for repairing them, and I'm going to talk first about the simpler of these two pathways, which is something called non-homologous end joining. This is a machine that only has 6 components, and if you imagine a strand of DNA that has just encountered a break, it undergoes chromatin modification. There's a modified histone called gamma H2AX, and there's deposition of sensors and transducer proteins, of which a protein called 53BP1 is an example. I think it would be helpful to have a pointer with a battery that's a little more active. The first step, we believe, in this machine is, thank you, is the binding of a protein called the Ku protein to the double-strand break. This is followed by the binding of the DNA-dependent protein kinase, the third polypeptide. It's one of the largest proteins in the cell, undergoes a very complex set of autophosphorylation events that remodel this complex, recruits accessory factors, and importantly, recruits a complex of 3 proteins that has DNA ligase activity and that reseals the break. So the first example that I want to give of what we're doing with this machine is modifying the components so that they can be tracked optically. So the neat thing about this repair process, and the reason that we were so interested in pursuing it, it's a focal process. In the light microscope, you can actually see these repair foci being formed and disappearing on a time scale of a few minutes. So we have 3 ways that we're tagging the proteins. First is quite a conventional way, which is a genetic fusion of individual components to green fluorescent, cyan fluorescent, yellow fluorescent, or red fluorescent proteins. We've done that, we've used those. The second 2 ways are more advanced, and they involve semi-synthetic approaches, one of which is putting small tags in the components that can be chemically labeled either ex vivo or inside living cells in various ways. And the third, which I think in the end will be the most promising, is the most exotic, is actually putting a nanocrystal on these proteins, a quantum dot. It's very stable, very bright probes. This is an example. This is a piece of DNA. That's a quantum dot. And then when these are mixed with a protein intermediate that binds the DNA on the one hand and the quantum dot on the other, you see these complexes of intermediate mobility, which we've not only now formed in the test tube and are not yet in cells. To study the assembly, why would we want to study this assembly and disassembly? Well, it's a nice thing to study. It's an important biological process. But perhaps more importantly, as we go to modify the function of these machines, we want to be able to see what we're doing, see how we've engineered them, see the result of what we've done. We need to induce breaks in a controlled way. We have several ways of doing this. One is enzymatic, but this is, I just wanted to show you something that we've invented together with Dr. Janata at Georgia Tech. When we irradiate cells, that's normally done in a $500,000 machine that has a lot of cesium in it and that requires a big fence around it and a security guards and health physicists to run it. It's very wasteful, right? You're irradiating all this air and all these culture dishes and all these places where the cells aren't. And we've developed a microelectrode. It's only 25 microns across, a little bit bigger than a cell. It's coated with a thin layer of isotope. The energy of that isotope is chosen so that the particles penetrate just a few microns beyond the end of the tip and eventually are arrested by the culture dish. And we can irradiate single cells one at a time. And using our bright fluorescent probes, we can collect real-time assembly data. And so this is a 1-hour time scale, repeated images of the same cell that has a microirradiator positioned just above it. And you can see these repair foci being formed.